Now we discuss about the infectious agents. Okay. So under this you have virus, bacteria, fungi and worms which causes different types of diseases. Now viruses which cause different diseases, under that you have common cold, influenza, AIDS, SARS. What is the full form of AIDS? Acquired immunodeficiency syndrome. SARS, severe acute respiratory syndrome. Okay. Swine flu, corona, poliomyelitis. Other than this you have mumps, measles. These are also caused by virus. Now bacteria, typhoid, cholera, then tuberculosis, then tetanus, diphtheria, then peptic ulcer also, acne, etc. Okay. Then fungi. Under fungi you have ringworm, and athlete's foot. Skin infection. Then protozoa also you have. Protozoa also. Virus, bacteria, protozoa are unicellular, whereas fungi and worms are multicellular. Okay. Then protozoa. Under protozoa you have Plasmodium. Plasmodium causes malaria. A vector is passing it, that is female Anopheles mosquito. Okay. Plasmodium. Then trypanosoma. Trypanosoma causes sleeping sickness, and Leishmania, causes Kala-Azar, Only 2-3 are enough. Now let us discuss about the worms. Some diseases which are caused by worms. Now worms like filariasis by Wuchereria bancrofti. This is also filariasis or elephantiasis. In this what happens? Leg swells like an elephant leg. Okay. Filariasis. Then you have ascariasis by roundworm or ascaris. Roundworm, that is ascaris. This ascaris is found in the human beings and other animals. Okay. Then you have taniasis. Taniasis is the name of the disease. Ascaris is the name of the disease. Filariasis is the name of the disease. It is caused by tapeworm. Okay. When uncooked pork is eaten, then taniasis may occur. Okay, that is caused due to tapeworm. Okay. Now let us discuss about the factors which causes the diseases. Okay. Now, as you know that these agents, that is bacteria, virus, these are microorganisms. Bacteria, virus and protozoa, they are unicellular, okay. So they have more or less, they perform similar metabolic activities. But when you take it separately, then the activities are totally different because their biochemical reactions or mechanism is totally different from each other. If you take only bacteria, all the bacteria will show the same activities, more or less. But when you study about the bacteria and the virus, two different factors or two different groups, then what you find? Bacteria, its function, biochemical functions or biochemical pathway that is taking place inside it, and the biochemical pathway that is taking place inside the virus, that is totally different, okay, different from each other. In bacteria, what do you find? They have a cell wall, they have plasma membrane and they may have flagella. Some of them may have or some of them may not have. But they do not have a nuclear membrane. For that they are under prokaryotes, okay. They are the primitive type of organisms. And this bacteria, so what is needed? The study of the organism is very much essential. For what? For the treatment. So that when you study about the organism, then only you can think of the treatment, how to treat that organism or how to control the spread of that organism. For example, if we are introducing antibiotics, what are antibiotics? Antibiotics are the medicines which control. Anti means against, biotic means life, bios means life. The materials which are against life. So in this you have penicillin was first discovered by Alexander Fleming from the fungus, that is penicillium. So what was found, this penicillin, what it did? It did not allow it inhibits the formation of cell wall. Where? In bacteria. So when bacteria was treated with penicillin, what was found? When penicillin was introduced in bacteria, when the bacteria are growing, when it was introduced, it was found that the bacteria cannot increase in number. Why? Because it was unable to form the cell wall, and cell wall is very important for the survival of the bacterial cell. So in this way, what happened? This antibiotic, it helped to control the spread of the bacteria or to control the growth of the bacteria. So like the penicillin was one antibiotic, and other than that, ciprofloxacin is one kind of antibiotic which control the DNA replication. Ciprofloxacin, it inhibits DNA replication. Okay. When this antibiotic is given, then it will not allow the DNA to replicate or increase in number. Then one more is there, streptomycin. Streptomycin, what it does? Streptomycin, it does not, it controls it is also an antibiotic which controls the growth of the bacteria. Okay, these are the different types of antibiotics. These antibiotics will act only on the bacteria. Okay, but if you get infection like common cold, and if you are taking antibiotic, will it work? It will not work, because common cold is caused by virus, and the virus is a completely different, it is a different group of organism, and its mechanism is totally different, that we'll discuss now. So this virus, if you get common cold and if you are taking antibiotic, it is not going to cure, because the common cold is caused by virus. Whereas along with the common cold, if you have some infection, bacterial infection in your body, then it may affect only the disease that is caused due to bacteria, but it will not cure the common cold. Okay, so specific medicine is effective only on the specific group of organism. So if you are taking antibiotic, it may control the bacteria of the other species also. Not only one type of bacteria, it can control the growth of the other bacteria also. Because more or less all the bacteria, they have the same mechanism. So it can control a variety of bacteria in your body. If for one type of infection you are taking, if some other infection is also there because of bacteria, that also it will control. But it is not going to control the infection caused due to virus. Okay, this is about the medicine. So the medicine that you use, that will control the spread of only a particular group of organisms or only a particular disease. Okay, it depends on the factor which is causing it. Now, in our body, what happens? Interferons. Interferons are developed. What it is? Interferons. When you get some viral infection, interferons. These are antiviral proteins. Okay. In our body, when you get some viral infection, naturally it develops interferons, that is antiviral protein. What it does? It controls the replication of virus. It will not allow the virus to grow. That is naturally formed in our body, in our immune system. When you get fever, that time more interferons are developed in our body. So it helps to control the infection. But if the virus is too much virus has grown, then it may not be effective. Okay. So interferons are developed naturally in our body. Now one more thing, earlier peptic ulcer, it is given in your book, peptic ulcer was considered to be caused due to acidity. But later on it was found that peptic ulcer was caused due to bacteria. What was the bacteria that caused peptic ulcer? Caused by bacteria. Later on it was discovered that peptic ulcer was caused by bacteria. Who? Warren and Marshall. Marshall, these are two scientists who found that peptic ulcer was caused by bacteria. Earlier it was thought that this peptic ulcer is caused due to the acidity, due to the lifestyle, when more acid, or due to the stress, when more amount of acid is liberated, and then due to peptic ulcer you have abdominal pain and sometimes bleeding in the abdomen or stomach. So when this scientist, Warren, what he did, when he studied the lower part of the stomach, he found that some bacteria are present in the human beings who were suffering from peptic ulcer. Then when he studied this bacteria and he grew and he did the culture of this bacteria, then he found that this peptic ulcer was caused due to bacteria, not because of stress or acidity. Okay. So later on Marshall and Warren, they got Nobel Prize also for this discovery, and later on they found that by introducing antibiotics, this peptic ulcer, it could be cured. It would take time, but it could be cured. Okay. So up to here today.